Hi everyone, welcome to S&S Bio Info. Today's topic is dihybrid cross. Dihybrid cross. Gregor Mendel conducted several hybridization experiments to study the inheritance of genes in garden pea or Pisum sativa. Garden pea or Pisum sativa. He conducted monohybrid crosses and dihybrid crosses to study the inheritance of one gene and two genes respectively. Monohybrid cross is a cross between two parents with one pair of contrasting traits, while the dihybrid cross is a cross between two parents with two pairs of contrasting traits. You know what is the difference between character and a trait? Character is a morphological, physiological feature of an individual, while the trait is an expression of the character. Gregor Mendel selected 7 characters and 7 pairs of contrasting traits of garden pea to study the inheritance of traits. The 7 characters are the color of the seed, the shape of the seed, the color of the pod, shape of the pod, the color of the flower, then position of the flower, and the stem height. These were the 7 characters selected by Mendel. The 7 pairs of contrasting traits are, in case of seed shape, round and wrinkled, seed color, yellow and green, pod shape, inflated and constricted, pod color, green and yellow, flower color, violet or purple and white, flower position, axial and terminal, stem height, tall and dwarf. These were the 7 pairs of contrasting traits selected by Mendel. Now let's learn how a dihybrid cross is done. I have told you that dihybrid cross is a genetic cross which involves the crossing of two parents. Crossing of two parents means cross-pollination of two parents with the two pairs of contrasting traits. Let us assume two parents. Then one more thing, that Mendel's experiment consists of 2 phases. The first phase is cross-pollination and the second phase is self-pollination. So let's consider one example, that is a round yellow plant is crossed with the wrinkled green plant. So round yellow are dominant traits, both are dominant traits, wrinkled green are 2 recessive traits. Then let's do the dihybrid cross now. Here the parents are round yellow and wrinkled green. So here round yellow are dominant traits, while wrinkled green are 2 recessive traits. So genotype. The next step is genotype. The genotype of the parent 1 and parent 2. Let us write now capital RR for round, then capital Y for yellow. So these two are homozygous pair of alleles because they are pure varieties. Here the next parent wrinkled green. So that is small RR, small YY. Then the gametes of each parent. Gametes. Parent 1, parent 2. So first one, let us assume that first parent is male, the second one is female. The first parent's gamete. Here for this case only one allele, one type of allele is present in one gamete. Again one from second pair. So these two are present in the gamete because as it is homozygous dominant allele, both dominant alleles are present in the gamete for controlling 2 traits such as the shape and color of the seed. In the case of second parent, so same thing, second female parent, the gamete receives one from each pair, one from each pair. A similar type of gametes are also formed in the case of second parent. So next step is fertilization. Fertilization is the fusion of male gamete with the female gamete. Here the male gamete with the female gamete. So what would be the result? Here the alleles that control the particular trait will come together. So let's write the R's together. Capital R from here, small r from the next gamete. Again the capital Y from one gamete, small y from this gamete. So what's genotype mean here? Heterozygous round yellow. Round yellow. All seeds are round in shape and yellowish in color. All F1 plants. F1 plants are first filial plants or first generation plants. This is the result of cross pollination. Cross pollination, the transfer of pollen grain from the male parent to the female parent. So this is a result of the cross pollination. That is the first phase in Mendelian experiment. The next step is the selfing of F1 plants. Here, selfing of F1 plants. So genotype of the F1 plant is heterozygous RY. So as it is a bisexual plant, both parents will have same genotype. So heterozygous RY. So the next step is gametes. Here there is a possibility of the formation of 4 types of gametes. Let's find out. Then here let's multiply this capital R with a small y. It's a 2 combination. So for 2 traits, 2 alleles must be there in the gamete. So that for shape, capital R, for color, capital Y. This is one combination. Second combination, capital R with a small y. Again, shape and color. Again, third combination, small r with a capital Y. Third combination. Then, fourth combination, small r with a small y. These are the possible gametes of the first parent. As the second parents, that genotype is also same one. So, same type of gametes are formed in the second parent. So, here I can write capital R Y, first combination, capital R with a small y, small r with the capital Y, small r with the small y. The next step is fertilization. Fertilization. Fertilization is a fusion of male gamete with the female gamete. So that we must combine the male gamete with the female gamete. They will get the genotype of the F2 plants. But by doing this, it's somewhat complicated. See, for that, a scientist named Reginald C. Punnett proposed a checkerboard or square to write the possible genotype of the F2 plant. That is called Punnett square. Punnett square. It was proposed by a British geneticist named Reginald C. Punnett. That's why this square is the box is known as Punnett square. So for drawing a Punnett square to find out the possible genotype of any generation F1, F2 or F3, we need to multiply the gametes of both parents. Here 4 into 4, 16. 16 boxes or squares must be drawn to find out or to write down the possible genotype of the F2 plant. Let's do how it is done. Here 4 columns, 1, 2, 3. Then 4 rows, 1, 2, 3, 4. Then total 16 squares. Here write the gametes of the male parent. Here the gametes of the female parents. So I write these gametes. Okay, first gamete, second gamete, then third gamete, then fourth gamete here. Again, second parent's gamete, female, that second parent, that is capital R Y. Again, the second combination, third combination, and a fourth combination. Now let's find out the possible genotype by combining with this one to these four, second one to those four, then third one to those four, and fourth one with those four. So first of all, this one. So when we write the genotype, the same letters must come together. Same letter means whether it is a dominant or recessive, the same letter should come together. So that I write here capital RR, capital Y. Okay. Then again capital R, capital Y, small y. Okay. Then again capital R, small r, capital Y, capital Y. Then fourth one, capital R, capital Y, small y. Then second one, here again capital R, then capital Y, small y. The next combination, capital R, small y, small y. Third one, capital R, capital Y, small y. So why I wrote that capital letter at first? Because whenever we write a pair of alleles, the dominant allele should come at first. So that I wrote so. So the fourth one, then capital R, capital Y, small y, small y. Again, these two, capital R, capital Y, capital Y. Again capital R, capital Y, small y. Third one, small r, capital Y, capital Y. Then small r, capital Y, small y. Then fourth one, here capital R, small r, heterozygous. Then again heterozygous Y. Next one, heterozygous R, homozygous Y. The homozygous R, heterozygous Y. Finally, homozygous RR, homozygous YY. So these are the 16 possible genotype of F2 generation. 16, total number 16. Okay. Here, let us find out the phenotype. These are the genotypes. So, then what are the phenotype of these plants? So, to write the phenotype, we need to consider the first letter of each pair. See, capital R here, again capital Y here. So, we can write the phenotype as round yellow. So, round, then yellow. Again, round yellow. Third one also, round yellow. Fourth one also, round yellow. Fifth one also, round yellow. Sixth one, round green. Round, then green. Next one, again round yellow, round, then yellow color. Then next round green, round, then green. Here again round yellow, then here round yellow, then wrinkled yellow. So for wrinkled I draw like this, wrinkled yellow, again wrinkled yellow, then next one round yellow, then round green, next one round green, then that wrinkled yellow, then finally wrinkled green, So now you can easily understand the different types of phenotypes here. 4 different phenotypes are observed in the F2 generation. They are, see, first one, round yellow. Round yellow. Round yellow. How many? How many? 1, 2, 3, 4, 5, 6, 7, 8, 9. Then number 9. Okay. Then next combination, round green. Here 1, 2, 3. So next round green, round, then green. How many? 3. Again wrinkled yellow. See, 1, 2, 3. So wrinkled yellow. That is also 3. So finally only one wrinkled green. Wrinkled green. Only one. So here the phenotypic ratio is 9:3:3:1. So total 16. 9 plus 3 plus 3 plus 1 is equal to 16. Here you can see both parental traits and non-parental traits. The parental traits are round yellow. The first parents are round yellow and wrinkled green. The last one is also the parental type. But these two are round green and wrinkled yellow are non-parental types. But there is no mixing of traits or characters in this case also. Gregor Mendel couldn't observe any type of intermediate or blending traits both in monohybrid cross and dihybrid cross. So that he confirmed that a pair of allele controls a particular character. In a dissimilar pair of allele, one allele dominates over the other. Then he proposed a law of dominance and law of segregation based on the monohybrid cross. But he proposed the third law, the law of independent assortment, based on the result on dihybrid cross. Here also there is no blending of characters, as alleles segregate independently at the time of gamete formation. During gamete formation, chromosomes segregate each other, then alleles also segregate each other, so that gamete receive any one of the allele for a trait. That is independent assortment. In a dihybrid cross, segregation of one pair of character is independent of the other pair of character. I hope you understood well what is actually the dihybrid cross. You can do the dihybrid cross with any two pairs of contrasting traits. You will get the same result as like this, 9:3:3:1. So you can easily find out the genotypic ratio. Then by seeing the genotype, how many homozygous are RR, YY, how many heterozygous round yellow, like that you can easily find out the genotype also. Then with this I conclude my session. Thank you and have nice moments.